comenzar con el seminario. El día de hoy tenemos invitada a la doctora Connie Weaver de la San Diego State University y la voy a presentar en inglés, así la doctora nos puede entender. Por favor, no se olviden de mutear los micrófonos, así no hay interferencias durante la presentación. Okay, so I'm going to introduce Dr. Connie Weaver. Dr. Weaver is a distinguished research professor in exercise and nutritional sciences at San Diego State University. She's a distinguished professor emerita of nutrition science at Purdue University, Indiana. She's an elected member of the National Academy of Medicine Science. She's a fellow of the American Society of Nutrition, Institute of Food Technology, American Heart Association, and American Society for Bone and Mineral Research. She's a member of the Science Advisory Boards of FDA, the California Prune Board, California Walnut Board, and she's the president of the American Society for Nutrition. And I had the privilege to work with her as a postdoc in this project that she will present right now. So Connie, our pleasure to have you here today.
Well, thank you, Gretel, for inviting me. It's a pleasure to meet all of your new colleagues and students there. I wondered if you had presented some of the work you did before.
Not with them, not with them.
Oh, okay. So I'll touch on some of that and mention where you were involved, but you can add in gaps. So she already told you my disclosures, and I would guess everyone on this call isn't involved in osteoporosis, so just a little bit of explanation for why bone health is important. Bone is like a bank for minerals, especially calcium and phosphorus, magnesium. And so if you don't get adequate supplies in your diet, your body resorbs bone to supply the blood with the needed nutrients. And so if that occurs chronically over time, the microarchitecture thins to the point you're vulnerable to a fracture. You can't really reverse this process. So I'm all about prevention rather than waiting till you have this state and try to then stop further loss. You really can't rebuild the bone. And consequently, there's staggering costs. In the US alone, there's 2 million fractures a year. The risk factors are low physical activity because you need that force on the bone to stimulate it, to tell your body you need the bone, and so that if you go into microgravity or bed rest, then you lose a lot of bone because that physical force isn't on your bones to stimulate its maintenance. Low calcium intake and vitamin D deficiency are the classic nutritional related associated problems that give you risk of osteoporosis. So the standard care for someone diagnosed with osteoporosis is to give them calcium and vitamin D supplements, because doctors usually don't have nutrition to advise them how to do it through diet, and participants aren't, or patients aren't that good at complying with their advice for diet changes. They're not all that good with supplement advice either, but they try through that route mostly. The non-modifiable factors you can't do much about. Genetics, if you're female, you're more at risk than males because 80% of hip fractures are in women. As you age, you're going to inevitably lose more bone and become more vulnerable to fracture. Caucasian, Mexican Americans tend to be more vulnerable than like African Americans. Half the hip fractures, that's where the real risk occurs, because you go into long-term care and 25% of those die within a year. But it's really not the morbidity that is so frightening for osteoporosis, because other diseases have a higher incidence of death, but the quality of life is compromised. And osteoporotic fracture is so much more prevalent than the other diseases that we spend more time worrying about it because of the higher mortality. Just to give you this proportion of how much more common fracture is. And we really ought to do something about it because the quality of life decreases greatly if you have a hip fracture. Then you can't socialize the way you would like to. Bone health is a lifelong concern. Contrary to what many people believe, since bone is slow to turnover, they think if you get to peak bone mass, then it's static. But that's not true. Bone turns over slower than other tissues. You get your peak somewhere by age 20 to 30, but then you want to hold on to the bone because it's going to remodel continually throughout your life. So men genetically have a higher amount of peak bone mass than females, and that protects them greater for fracture, but obviously not completely because 20% of hip fractures are in men. So we want to prevent fracture by building up the most bone we can during peak bone mass development, and then to hold on to the bone as well as you can later in life. Well, kind of the new area for investigation is what is the role of the gut microbiome? And it's completely intertwined with diet, because you need the substrates to feed the lower gut microbiota. Usually we think of that as being dietary fiber, which then is fermented by the fiber fermenter type of gut microbiota to produce short chain fatty acids like butyrate. And this can have a direct effect on bone metabolism, but through osteoclast precursors, or indirectly through immune regulation, through TREG regulation, absorption of minerals like calcium, and endocrine modifications like IGF-1 modulation. But it's not just the fiber we're finding, it's metabolites from other sources of the diet, like blueberries. We're going to be talking about that. So blueberries are a really good model of polyphenol-rich fruits. And of the dietary factors that are being studied related to bone, polyphenol-rich fruits are among the most studied, and they also have fiber. So it's a rich source of polyphenols, especially flavonoids. It's among the highest phenolic content of commercially available fruits and vegetables. And so there's some dialogue around maybe qualifying the dietary guidelines in America, at least, to think about recommending polyphenol-rich sources. So we have a little precedence of that with citrus. We say, be sure some of your fruits are citrus to get vitamin C. Wouldn't it be something if we got a little bit more nuanced and said, consume polyphenol-rich fruits because of their advantages on immune system and bone? And so the committee has to decide if there's enough evidence to do this yet. But there's a rising popularity of polyphenol-rich fruits, and likely the most popular one is blueberries. So as Gretel alluded to when we were chatting in the beginning, we were collaborators on a big NIH grant that lasted from 2014 to 2019 and is actually still going on a little bit. The pandemic disrupted a lot of the completion for a while, as it did so many other projects, but we're mostly published this work now. But here are some of the main co-investigators that worked on the project, and it represents a very diverse set of expertise. So you have nutrition, statistics, mass spectroscopists, food scientists. You have the plant physiologist who really studies blueberry cultivars with Marianne Lila. You have bioavailability experts. Here's Teresita Bolito, who was the mentor for Gretel for her postdoc, who's really well known in animal studies and antioxidant behavior, along with David Burr and Monroe Peacock, who were all at IU School of Medicine during the time Gretel was there. And Monroe Peacock has been the clinician with the human studies I've been involved with for decades. He's retired now, too. So we took Marianne Lila's work in North Carolina, who had just profiled the anthocyanin and other polyphenol content of hundreds of lines of blueberries in their genetic biobank, if you will. And so that was really handy at the beginning of this grant, that we said we knew the profiles of like 1,200 blueberry lines. So blueberry is not a blueberry is not a blueberry. They can be blue or white or green to give you just sort of a superficial idea. But in order to pick which ones to pursue in further research, our statistician did a principal component analysis and picked spots on this principal component analysis that were most different in polyphenol profiles. And then we picked ones that could be grown to get a substantial amount in cultivars that were very close to these circles to separate the principal component analysis. So we picked 6 genotypes to further study: Ira, Montgomery, Onslow, et cetera. And you can see different ripening stages in this picture, but some end up looking whitish or light green even at the maturity. And we did bioavailability studies in rat models especially. So this takes these different varieties that we picked out plus the commercial mix that was available. And this just shows you one bioactive metabolite of them, showing you how different they are. So Montgomery had the most of myristoyl glucuronide of any of the varieties, and this SHF2B1 had hardly any. So we did dozens of these areas under the curve for different phenolics, showing many differences. So it wasn't always that Montgomery had the most of a metabolite, but it did some key metabolites. And you'll hear Montgomery is one of the most successful ones in terms of bone. And this is probably the explanation for why. So the goal was to determine the effective blueberry dose for increasing calcium retention in OVX-rats. And this was one of the earlier studies after we did the bioavailability studies to look at what is the dose that makes sense. So what was done is we pre-labeled the bones with a calcium tracer of rats. And then they went through a randomized treatment of 10 days at 2.5% of the diet, 5, 10, or 15% of the diet. And here's the design. So you first give them a dose of radioactive calcium and wait a period so that the soft tissue tracer is flushed out and all the calcium tracers in the bone. Then you collect 24-hour urine collections periodically to see how much calcium is coming out of the bone, because after the soft tissue tracer is flushed out, all the calcium that's left of the tracer is in the bone. So any tracer that comes in the urine tells you how much net loss of bone there is, because calcium is a constant fraction of bone. So you get a baseline measurement, and then you start giving different interventions of these treatments, and you collect the urine throughout, and you determine how much calcium retention in the bone there is. So this shows you the dose response results of that. The dose that was significantly superior in retaining the tracer was the 5% blueberry diet. 2.5% wasn't bad. It was almost significant. Another rat in there would have been significant, but the 5% was significant. And if you got to higher levels, it was no longer significant, suggesting a hormesis effect. And I'll talk more about this later. Then we did microbiome analysis on that study, and it showed you that the principal coordinate analysis of the microbiome separated pretty well by dose. So you have the baseline in red and the 2.5 and the 15% blueberries separating out cleanly from the baseline where there was no blueberries. So the gut microbiome switched. Now, I'm not going to elaborate much on the study that Gretel was involved with Teresita Bolito at IU School of Medicine. I'm just going to give you the summary of many months of work with OVX and ORX, male and female mouse models. And so both female in red and male in blue are thought to have the same issues as they age and go through sex-steroid hormone deficiencies, naturally through menopause with women, more gradually with men, but we introduced sex-steroid deficiency by surgery. So OVX in the females and ORX in the males. That leads to increased reactive oxygen species production, which stimulates osteoclasts, the cells that chew away at bone and increase resorption, and apoptosis of the cells. So it happens kind of in the same pathway, and that results in bone loss unless you intervene. So we studied mechanisms like if it was NRF2 dependent or not, and the group, Gretel and her colleagues at IU School of Medicine, measured endogenous antioxidant gene response, and in response to berries or not berries, the controlled diet, and found these remarkable sex differences. So in the females, the mechanism was through NRF2, and berries could completely stop this process that leads to bone loss. So it was very effective. But in males, not only did it not work through the same mechanisms, but berries could only partially recover the bone loss due to the surgery. So huge sex differences, and it reminds us that sex is a biological variable, and we ought to be considering that when we design studies. Now to the human study that was informed by all these animal projects that we did, that was just published last month in the American Journal of Clinical Nutrition. And the lead investigator was Kalina Hodges, who was a postdoc at that time, and now is on faculty at Penn State University. So she used calcium 41 to label bones of a human, much like Maria labeled the rat bones I talked about a minute ago, but calcium 41 has a very long half-life. So once you dose a person with calcium 41, you can follow bone loss in the urine for years, mostly for the rest of their life. And because postmenopausal women is the most vulnerable group, we studied postmenopausal women only and did a dose response, low, medium, and high in randomized order. So the low dose was equivalent to 1/2 cup of blueberries. So we used whole freeze-dried blueberries. And the medium dose, 1.5 cups equivalent, and the high dose, 3 cups equivalent. And we did a lot of product development to see how we could get these whole freeze-dried blueberry powders incorporated into a palatable source of food to give to them. So we developed a spread a granola bite and a juice, a drink, and they could pick any two of these three to consume 2 servings a day. So here's the design where we dosed them with the calcium 45, waited 150 days for the soft tissue tracer to leave the body, so it's all in the bone. Then we randomized the order of low, medium, and high dose with washout periods in between and one at the end. So the comparator are these non-treatment periods against the treatments that we studied with blueberries. So to digress a little bit to tell you the power of this method with the calcium 41 tracer, the traditional way of looking at an intervention on bone is to look at bone mineral density through DEXA measures. But because bone turns over at a rate of 26% a year for trabecular and only 3% for cortical bone, then the FDA approved study design for determining efficacy of like a drug type intervention would be a 4-year randomized controlled trial of bone mineral density. And typically it's hundreds of participants are required. So the calcium 41 allows us to develop a 42-day screening method because it is so sensitive relative to DEXA. And then because it's short, we could do multiple comparisons in the same people, which gives us a huge amount of power. So for 12 people, we have the same power as that FDA type of design that for hundreds of people. So the rare isotope calcium 41, along with these other rare isotopes, are measured on an accelerator mass spectrometer. This is a giant machine. The whole thing is the size of a football stadium underground to reduce background, but it can measure like atom quantities, 10 to the minus 18th molar, and it's the power of the measurement and the long half-life of calcium 41 that allows it to be so sensitive. So here are the results of that postmenopausal woman study. Like the rats, the lowest doses were the most effective. So 3/4 of a cup a day equivalent was the best. Cup and a half was also significant, but by the time you got to 3 cups a day, it was no longer statistically significant with much more variation, thinking that we had saturated the transport mechanisms. So that made us think, what are people doing out in practice? If they're taking supplements, you can get a lot higher levels than you can get with foods. So there are blueberry extract supplements, for example, and taking this increased use, we thought, well, could there be harm with blueberry supplements? So Dennis Cladis, my PhD student at the time, was exploring hormesis against the non-hormesis essential nutrient curve. So hormesis is something that happens, you get into adverse conditions with increasing dose. And we were starting to learn when he did this from the rat study that the higher doses were not effective for bone. So he was really asking the question, can it be harmful? So he posed high doses of blueberry polyphenols may overwhelm normal metabolic process and alter GI function. So unmetabolized flavonoids, if you saturate the transport mechanisms, could lead to altered gut function and decrease calcium absorption or increase it, either way. So he did a 90-day toxicity study for the safety of and studying metabolism. So 90 days is a classic toxicity type study to look at these higher doses that you can get with the extract, and looked at what was going on with the GI tract. And he hypothesized higher doses might be toxic, And the metabolism may increase at the beginning of the study due to gut microbiome adaptation, but then would plateau and stay constant. So here's the design of this 90-day study. He stabilized the animals after OVX and did a daily gavage with control, low, medium, or high doses of blueberry. And these are as the extract. And then he gave blueberry powder dose at the low dose equivalent to see if the matrix also mattered, as well as the dose of polyphenols. So he did a systematic approach to assessing safety, the classical toxicity type of outcomes, and looked at clinical findings, survival rate, body weight, food composition, clinical pathology like hematology, serum chemistry, urinalysis, and looked at individual tissues, gross necroscopies, tissue weights, histopathology, intestinal permeability, and bone mineral density. In short, blueberry polyphenols were found safe. There were no biologically significant changes in any of those parameters. So he proposed, based on his data, that you could propose a NOEL of at least 1,000 milligrams of polyphenols per kilogram body weight per day. So that's a human equivalent of 10 grams polyphenols in 70 kilogram person. Meaning you couldn't get there at all by diet or even by the extracts in any kind of practical sense. So they're safe. When he looked at polyphenol metabolism, he found it's a complex, multiple step process. So first, in the upper gut, you're looking at bioaccessibility, which is the release from the food matrix and getting the absorption across the intestine. So the parent polyphenol could get across, phenolic metabolites could get across. But in the lower gut, you have the action of the microbiota, and they can produce phenolic acids that can get across the gut in the lower gut. So by collecting the stools and looking at these metabolites and what might happen when these metabolites go to the liver from either the upper gut or the lower gut, then he found phenolic acid metabolites starting to show up. So you read about bioavailability is very poor from polyphenols. So the upper gut is less than 2%, very low. And even with the gut microbiome, it's still only 15 to 20%, but that's a big leap forward compared to absorption from the upper gut. So he looked at a lot of individual polyphenol metabolites through this 90-day study, and he showed a dose response in all of these metabolites mostly. So this shows you just a sampling of some of the metabolites. And to summarize what's happening, there's an interaction between the polyphenols, their metabolites, and the gut microbiome. So you have substrates getting to the gut microbiome from the host metabolism of polyphenols, plus the metabolites produced by the gut microbiome. And these can enter phase 2 metabolism. He looked at tissue levels of these metabolites, and there was little evidence of long-term storage. So he detected a total of 15 metabolites, 6 of them he could quantify. And in the small intestine, colon, liver, kidney, and brain, here's the numbers of metabolites. 3-hydroxy PPA was quantified in all the tissues, hippuric acid quantified in all but the brain. There was little evidence of any tissue storage, most likely excreted within 24 hours. So quite a rapid turnover. So looking at the gut microbial diversity affected by dose and the food matrix for alpha diversity, the thin sample was evidence of a hormesis where you have, let me get the code up here. You have blueberry is different from the control, and you have the medium dose and the high dose relative to control. So the high dose is really different than the lower doses. And then the beta diversity was dose dependent, much like I showed you with the earlier rat study. So contrasting the high and the lower doses. So diversity changes in high dose were driven by taxa dominance and taxa extinction. Blueberry polyphenol metabolism is dependent on dose and the food matrix, he discovered. There were shifts in the gut microbial populations as the polyphenols reached the gut microbiome and they were metabolized. The phenolic metabolism is saturable and dose dependent. There's extensive host xenobiotic metabolism. And there was a matrix effect. So blueberries were not, polyphenols were not found to have the same effects as the blueberry extract. I wanted to show you a little bit of the other kinds of work that we've done, saying it just all isn't about blueberries. Blueberries are very popular, but we recently reported a study in prunes. So prunes are another high polyphenolic rich fruit. And this study was, the PI was Mary Jane D'Souza at Penn State University. And it was a classic type of study, a one-year intervention with hundreds of people and followed by DEXA changes. And it was also a dose response in a parallel arm design, control 50 grams of prune a day or 100 grams of prune a day. And the exciting results were at the total hip. So you had a benefit of the prunes on lowering total hip compared to the control. The control, the women lost bone. These are in postmenopausal women also. They lost bone as is typical with postmenopausal women, but the 50-gram dose and the 100-gram dose could prevent that loss. So the total BMD is on a percent change. This shows you the loss of the control and either dose. And we had more dropouts with the 100-gram. That was a lot of prunes to get in the diet. But 50 grams is like 5 or 6 prunes, and they could tolerate that better, and it was just as effective. Another source that we spend a lot of time studying, this one shows you in postmenopausal women, but we've also had 2 studies in adolescents. This shows you, using our calcium 41 technique, the dose response advantage of retaining bone in postmenopausal women that otherwise would be losing bone. So soluble corn fiber is something that's in a lot of supplements that you can get on in most major retail stores, or you can order it online. It's very inert. You can sprinkle it over foods or stir it into beverage, and it's pretty tasteless and you don't even notice it's there, but it's a fermentable fiber for the gut microbiome too. So from our 3 trials that we did that were positive for bone, FDA guidance said soluble corn fiber meets the dietary fiber definition on the nutrient facts label based on our calcium absorption and retention studies. And so that was a big accomplishment. It also stimulated other trials, a 1-year randomized controlled trial in children in Malaysia, and another 1-year randomized controlled trial in Florida with Christina Palacios as the PI, and that one's still underway. It led to a randomized controlled trial in bariatric patients with Ann Schaefer at UCSF, and to a study in type 1 diabetic mice that showed a real advantage in protecting against the bone loss associated with type 2 diabetes. So now I'm going to shift gears a little bit from research results to work I've been associated with in developing policy for how to approach it. because policies around non-essential nutrients have really lagged behind essential nutrients. So this group worked for a couple of years and came out with this publication in 2021 for a framework for how you would evaluate evidence to say, could you make public health recommendations for these non-essential constituents that have bioactive health properties? We argued that a framework for bioactive intakes is needed because without evidence-based recommendations, consumers utilize information from unreliable sources on labels in the media and whatnot. The framework was developed to be used by credible national and international health and government organizations that are setting recommendations. Recommendations require at least moderate quality of evidence in order to have a recommendation we proposed. And it was to help researchers design rigorous studies on the relationship of bioactives to health to say, here's what it would take to have a strong enough evidence to lead to a public health recommendation. So what health outcomes are used? Causal relationships supporting normal structure or function, maintenance of health, or reduction of risk of disease. Examples that we listed in this paper include maintaining or improving postprandial blood glucose, macular pigment density, cognitive performance, blood pressure, and bone mass or density. And here are the basic steps of the framework. The first thing is to characterize the bioactive compounds. And that's the sticky wicket for a lot of these things. We don't know what the bioactive is in blueberries or prunes, for example. Ensure sufficient food composition data are available. How much of these polyphenols, if we identify which ones are combinations or the dietary fiber is available. Determine that the intake is quantified by reliable intake exposure or a validated biomarker. And then evaluate the safety, kind of like what we did with the toxicity study with blueberries, or documentation of its history of use in a long-term population, for example, and ascertain intake with no known adverse health effects. Then conduct an efficacy review, select a relevant health outcome, and identify the specific indicators of physiology or biochemistry and conduct that systematic review of health relationships. Then forward, if you can accomplish all that, you decide on the recommendation. And as I mentioned before, it's got to have at least a moderate quality of evidence or don't make a recommendation. And we propose an intake range rather than a specific value, the way they usually do for essential nutrients. So here's a proposed statement based on, and you would say either moderate or high, depending on the quality of evidence. The recommendation is made to consume between X and Y grams daily of whichever bioactive or functional food to, and then relate what the outcome that you're talking about is among the general population. Now, this stepwise process has been applied to one class of bioactive compounds so far, and that's flavonoids. And so that's been published. And it's the first one through, and there's a lot of interest in taking other bioactive compounds or foods through. So in summary, a rapid method for evaluating efficacy of interventions, the calcium 41, showed promise for middle doses of blueberries in rats in postmenopausal women. And there's evidence for a hormesis effect, meaning it goes away if it gets too high. The sex differences in bone in response to blueberry feeding and mechanisms were observed in NERF2 mouse models. Extracts of blueberry polyphenols were found to be safe in human equivalents of up to 10 grams polyphenols in 70 kilograms a person. And the process and policy have lagged behind discoveries with bioactives, but a DRI-like framework was developed and published in 2021 that will be very useful going forward. So with that, I'm happy to take questions.
Thank you, Dr. Weaver, for your wonderful talk. And it's impressive to see how from the anecdotal of saying blueberries are good, we are going to a public health policy and saying more or less how much we should intake to see benefits. So that's amazing. So I have a question since I've been involved. So we noticed the dimorphism between male and female in the antioxidant response.
Right.
With the blueberries. So I'm wondering, I don't remember if you extended this study only to women or to men as well, and if you've seen that in humans.
So that's exactly the topic of the grant that I was trying to take this work further. We only did postmenopausal women. When we wrote that grant to do that study, we didn't know there was a sex difference yet because you hadn't done that mouse model study in humans. But it's really important to do, I think, in humans, the male versus female. And we were also proposing in sex-steroid adequate condition and deficient condition, because there may be an interplay, most likely an interplay between diet, sex, and sex-steroid status. But I haven't got the funds yet to do that. Maybe someday.
Great. ¿Alguna otra pregunta? Si quieren ponerla en el chat. Okay, Dr. Seni está aquí.
We have not studied the blackberry in secondary osteoporosis like diabetes. That's one question I saw in the chat. There has been some animal work with different kinds of berries, but keep in mind the study that we published last month is the first human study with berries and bone of any kind. We have the prune study that was published a few months ago, but the work with humans is barely almost non-existent in terms of randomized controlled trials. There's been epidemiological studies that show fruit and vegetable consumption is beneficial to bone, and polyphenols in general are. But in terms of randomized controlled trials, there's a little work with prunes. There's been a couple of labs, 3 labs, that have shown benefits of prune to humans. But we did the first blueberry, and I don't know of any other berry human study.
And Dr. Weaver, I don't know if you want to comment on the variety of blueberry. When I started working on your project, I didn't know the amount of varieties of blueberry that we have.
Yes, and I meant to mention that when I showed that overview scheme from your work, but 2 different blueberry lines were studied in that trial, and Montgomery, that had that high bioavailability of the myrcin metabolite that I showed you, was very effective, but another one, Onslow, was not. And so we really need to know, because in order to recommend blueberries, you have to do it on something about the blueberries that are effective. And it's a very complicated situation.
Well,
is there any reason to explain the differences by sex? Well, Gretel probably could do that better than me, but that one schematic showed females worked through a NRF2 dependent schematic, and the antioxidant gene response was strong in the females and not in the males. So that's a clue of the different mechanisms that are involved, but why they get that way, I don't know. Do you want to add anything to that, Gretel? You're on mute.
They have a question.
One
No,
second.
the difference from the consume of polyphenols by sex.
If they should consume more.
Yes. Yes.
Okay, so I think one more question. I think Dr. Seni wants to ask whether male or female should consume a bigger or higher or lower amount of polyphenols.
Yeah.
I mean, if there's any difference for male or female in the dose, let's say, of polyphenol that they need to consume.
Well, we don't have the answer in any human data at all, but Gretel's paper in JBMR would suggest it didn't work completely with the male at all. So I would say go for a different kind of strategy for male osteoporosis. And the women responded well to the berries, but not so well for the males. So I don't know that there's a dose to recommend for males. With the current evidence, I wouldn't put much hope in berries for male, I guess. So somebody was
and there's one more question in the chat
about the microbiome. I was
exactly.
I was looking about that, and there are differences, and you'd have to go to that paper to outline them, but there's not an obvious answer like you're proposing here, this increase in bifidobacterium or something. Nothing jumped out the way it did with the soluble corn fiber study that I mentioned briefly. In that case, more classic bacteria that you might think, bifidobacteria, increased with soluble corn fiber fermentation. But with blueberries, it's a lot more complicated because it's not only the fiber, but it's the polyphenol metabolism. So we saw differences, but you couldn't point your finger at, oh yeah, that change in the gut microbiome is the explanation for all of this, the way it seemed to be for the soluble corn fiber. But we didn't do any, we just did 16S microbiota. We didn't do any of the pathways. And I think you would have to do that, the metagenomics, to get at what was going on with the blueberries, really. And children, no, they haven't really been studied at all. It's rather a new field, actually. There's a lot of opportunities for y'all to design more research. So there's
there are two more questions.
There's some in Spanish up there. If you do you have, my Spanish is too rusty. Do you see that one? Si tienen preguntas pueden hacerlas. What did that say? What's that?
So
asking?
I think there are two more questions. I don't know if you get to see them in the chat. One is the consequence of one component in particular, or a synergy of different molecules in blueberries.
Well, I was thinking more, it's the synergy between the host metabolites in the gut microbiome metabolites. Yes, probably there are synergies within both of those, but kind of the big finding was it's not just the gut microbiome, or it's not just the human metabolism, it's the interaction. And there's hundreds. of compounds, we could quantify like 12 or something, sort of small numbers, but it opens your eyes to the complexity of what's going on. As our techniques evolve with omics, we're going to learn a lot more than we know now. This is sort of scratching the surface early.
And there's one more. Is there any suggested dose for children?
Yeah, children just haven't been studied at all. We don't even know if there would be any benefit to people who have adequate sex-steroid hormones. In other words, premenopausal women or males before they've lost a lot of androgens. That's why I wanted to study them and proposed in this grant, but so far I haven't been successful in getting them.
Okay, here are more questions. Are there any relationship between polyphenols and vitamin D and calcium?
Yeah, we don't know that because we worked really hard at keeping calcium and vitamin D constant through and adequate throughout all these studies we designed to take that off the table. I think if you're deficient in calcium and vitamin D, then correcting that may be more powerful than adding in the berries. That's what happened in Malaysia in that children's study with soluble corn fiber. They were so deficient in calcium. We studied soluble corn fiber with and without calcium. The calcium had such a dramatic impact on improving bone. You barely could see anything from the soluble corn fiber because they were so deficient in calcium. I think that could work here too, is you got to correct the calcium, the biggest constituent of bone mineral matrix first, before you can see more subtle advantages of the polyphenolics. But the polyphenolics don't just work on bone, they work on cognitive function and immune function as well, which calcium doesn't. So I don't think it has to be either or. Get your whole diet in good shape,
Did you evaluate?
And then another question is, did you evaluate the effect on other type of metabolism, like for example, fat, energy?
So you did in your mouse study, you found a pretty big advantage for energy metabolism. Were you involved with that?
No.
Part of it? Okay, so I know Teresita Bolito was even more excited about that than the bone, because it seemed to control the OVX-induced weight gain that you typically see, with the loss of estrogen, and the berries helped control that weight gain. And she thought that was maybe even more exciting than the bone protection.
Okay, let me check. Okay, so I think we don't have more questions. So thank you very much.
You're
Dr. Weaver,
welcome.
for joining us.
Thanks for having me.
Thank you.
You bet. Bye, everyone.
Bye.
Thank you so much. Have a good day.
You too.